We don't control the.
Can you hear us now? Zoom attendees, can you hear us?
Oh, yes, now.
Yes or no?
Oh, yes.
Okay, thank you.
Perfect.
Thank you.
I haven't said anything important yet. So these neurons are kind of like an electrical circuit, right? Where they transmit electrical signals, and the electrical signals associated with neurons are these depolarization events. However, unlike an electrical circuit, these neurons don't physically necessarily touch each other to transmit these electrical signals. So something else has to gate that information transfer, and that's chemistry, right? The chemical efflux from one neuron that will then either activate or deactivate downstream neurons that essentially have receptors or ears that can hear these signals. And the types of chemicals are various, right? So there are different chemicals the way there are different languages, and not all neurons speak all languages. Some neurons only communicate with certain neurochemicals, and they can differentiate between them the way we can tell the difference between something like English and Spanish. And so the goal that we have is very specifically to develop a method to be able to image these neurochemicals selectively, and then implement them to be able to understand how it is that neurons will, again, exocytose these neurochemical signals, how largely they propagate, how numerous they are within these tissues, and what that says in turn about health and disease. Now, to motivate the importance of this, and for those of you in the room who kind of understand kind of neurobiology and the tools that exist to image these types of signals, you'll know that there are many options, right, for kind of understanding how neurons communicate. And these electrical signals have been the focus of neurochemical signaling for some time. However, these electrical signals are akin to the activity of the cell, but don't necessarily correspond to what is being said or what is being spoken, right? So it's kind of like this scenario that I'm sure we've all experienced in the past few years where you're on Zoom and you're trying to communicate something, right? But you're on mute, and so you don't actually get any signal across. So I don't know if anybody in the room, show of hands, who's had this experience where you've been trying to speak, but you're on mute. Oh yeah, so like 90% of the room, and then 10% are just basically lying. But this is, I hope, a relatable experience, and highlights the importance of, why it is that we would want to be able to image the chemistry directly, because it's very much feasible and probable, and in some disease cases especially, conceivable that neurons might be physiologically behaving as they normally should in terms of their electrical activity or basal metabolic activity, but unable to efflux this information. All right, and so now focusing a little bit more on this chemical activity, as I mentioned before, we're largely focused on a class of neurotransmitters known as neuromodulators. The reason we're interested in neuromodulators is because they are thought to not only exocytose from the presynaptic neuron, but also diffuse outside of the synaptic cleft to modulate broader networks of neuronal activity. And these kind of volume transmission components of neuromodulation have a lot of implications in how receiving cells will then modulate their sensitivity to neurochemical signals and drive much more complex behaviors, including things like learning, pure bond formation, and things that are deviant from that, including things like addiction or social disorders. So dopamine, norepinephrine, serotonin, and neuropeptides that I'll talk about later are all kind of implicated in this kind of complex behavior. And so those were some of the targets that we were most interested in going after when we started the lab. Now, aside from the neurotypical behaviors that these chemicals drive, of course, aberrations in these neurochemical circuits are known to drive a lot of disorders, right? So dopamine and aberrations in dopaminergic signaling are thought to underlie addiction and schizophrenia. Aberrations in serotoninergic signaling underlying ailments like depression or anxiety. And more recently, though this has been hypothesized for a while, molecules like oxytocin driving autism spectrum disorders. And at the spatial and temporal scales of relevance, right? So the fundamental units of information transfer, which is on a synaptic scale, these signals have been largely invisible until very, very recently. And again, a lot of the hypothesized mechanisms for their central implication are based either on optogenetic manipulation of neurons that manipulates the activity of the neuron, but perhaps maybe or maybe not the chemical activity, or on studies that look at basically neuronal cell health within these circuits and derive that these disorders are coming from basically cell death or aberrant cell metabolism within these networks. And so the approach that we've taken is one that is based on a semiconductor known as a single wall carbon nanotube. So this is a one-dimensional carbon nanomaterial that in this particular isoform develops a band gap, hence being a semiconductor. What this means is that you can take a laser and excite an electron from the valence to the conduction band of the material, creating an electron hole pair or an exciton. And that exciton will recombine to do one of two things. It'll either provide a phonon or a photon. And this ratio of photon to phonon generation is very, very sensitive to the environment directly surrounding the carbon nanotube. So this makes this fluorescence source really nice for chemical-based imaging or chemical detection, because you can play with this ratio of photon to phonons by basically modulating the surface, the dielectric environment on the surface of the carbon nanomaterial. Another reason we like to use these probes is because a lot of our studies, yes, Paul.
How long is
How long? They are about a nanometer. I think the dimensions are on our next slide, but a nanometer wide and then on average a few hundred nanometers long. Yeah, it's polydispersity on the length axis. One of the reasons we like to use these probes is because they're quite photostable. So a lot of our measurements are taken over the course of an hour or more. And so we need the photostability that these semiconductors provide. And they fluoresce in what's called the tissue transparency window. It's not really transparent, but this is a window where photons are, I'd say, minimally perturbed by the absorption of biological fluids like blood, and right before water starts absorbing photons. So we can take images basically several tens of microns deep into tissues without any optical interference from the brain tissue itself. Okay, so that takes advantage, or that I guess explains the optical advantages of using these probes, and then how we make them chemically sensitive is, is a whole other question. And compiling close to a decade of work in just a couple of slides. But basically what we do is we will functionalize the surface of these nanomaterials such that the exciton recombinations predominantly favor phonon generation, right? So this material is very dark in this state here. And then we make this polymer perturbable only by a specific analyte, like dopamine or serotonin or oxytocin. So the analyte perturbs that polymer, that dielectric environment, causes a change in exciton recombination, either frequency or at least the frequency of photon generation, causing an increase in the fluorescence. We're working currently with a multi-chirality spectrum just based on how these materials are synthesized, which means that we have all sorts of different colors of nanomaterials, and we're taking integrated signal over all of those colors when we use these probes in tissues. Okay, so one of my very first students, Abraham Begane, who's now a group leader at Janelia, developed the technology. So he took on this project and hypothesized that some of the generic kind of ligand-based interactions that I'd worked on as a postdoc could be used to detect a molecule of neurological relevance like dopamine. So what Abraham did is he looked at the brain region in which we wanted to eventually do biological studies, in vivo studies, and looked at all of the neuromolecules that are found in what's called the striatum, right, the motor center of the brain, or the basal ganglia, I guess, a little bit more broadly defined. And so he developed a library of nanosensor candidates, basically just guessing what polymers might attach to our target molecule, which was dopamine, and then also included separately in the analyte library all of these other neuromolecules that are found abundantly in the basal ganglia of the brain, right? So that when we get a modulation in the nanotube signal, that we could iterate towards selectivity and sensitivity needed to then use this probe to image endogenous dopaminergic signaling. So again, just very quickly going over many, many years of work, the bottom line is that we were successful in identifying one of these polymers, specifically a repeat of GT nucleic acid, single-strand DNA, that provides an over 2,000% modulation and fluorescence upon addition of dopamine ex vivo. So then what we wanted to do was see whether or not this probe could be used to measure endogenous dopamine signaling in living brain tissue. And so this is a collaboration that started with Linda Wilbricht in the Department of Psychology. And Linda studies dopaminergic signaling as it pertains to adolescent development. And so what we did with Linda's lab was we took mice, we extracted brain tissue, from mice, and then we generated coronal slices of about 300 microns thick. So this really nicely preserves basically the architecture of neuronal circuits in the brain tissue, and we can keep it alive by incubating that tissue in artificial cerebral spinal fluid and bubbling it with what's known as carbene gas for several hours. So that provides us with a nice window for experimentation. We can then label that brain tissue with our infrared dopamine sensitive probe by co-incubating it. And so what we do is we flood the extracellular space with this probe so that we're not looking at the cells themselves, but rather we're looking at the void space. We're looking at a labeled extracellular space where we can take measurements of how these chemicals are being essentially effluxed into the ECS. All right, and so what our data look like are this, right? So we have this labeled tissue before stimulating the brain tissue. We have a dark, mostly photon generating material. We can stimulate either with optogenetics, so channel rhodopsin expression into dopaminergic neurons, or with an electrode, single pulse electrical stimulation, where we get a signal that shows dopamine efflux from cells into the extracellular space. And then through a combination of both diffusion and reuptake by dopamine transporters, because dopamine is recycled in neurons, we get this decay, which we can fit to a decay constant and use for, again, downstream analysis. But this is essentially what our data look like, right?
Markita,
Yeah.
Are these your scale bar on that?
So this is 10 microns. Yeah, yeah, good catch. Yeah, so we're looking at release sites on the order of a few microns.
And do you know how well you're getting the nano whatever into it?
So we have not done this work, but there's quite a bit of work by scientists in Bordeaux, Laurent Grenier, who uses single molecule imaging to track individual nanotubes to map the ECS using basically just using diffusion. So he's able to map out both the extracellular space and also the synaptic space. So these high acid duration materials will diffuse into the synapse. And because there are these very, very long tubes, the diffusion is slow enough to where that can be tracked and used to basically reconstitute the three-dimensional volume. So here, I mean, obviously this is a diffraction-limited epifluorescence image. So here we're looking with resolutions that are on the order of infrared wavelengths divided by 2, at best 500 nanometers. But with our control experiments, we're confident that we're labeling all points in the ECS. So we're not creating these pockets of signal due to the absence of probe. All of the void space that we see here is the presence of cell bodies. Yeah, but in theory, we are getting signal both from the synapse and from the volume transmission extrasynaptic efflux of the chemical.
Just to follow up on that line of questioning, so are you able to also do visible fluorescence or bright field to actually identify where the cell bodies are and see that you are seeing the signals from the interstitial?
That's right. In theory, we can, and we have done the experiment where separately we image in the visible spectrum, and we label usually only dopaminergic cells or cells of the neuronal type that we're imaging. Otherwise, it gets very crowded. It's a 300 micron thick tissue. But I'll show one slide on data from Abraham's lab doing this in two-dimensional cell culture, where those signals are much easier to cross-correlate. So I don't have the confidence in how the microscope is currently set up to say this signal is coming from this cell. But what we can do is take a look at the tissue and say there are dopaminergic cells there. All right, and so in kind of movie form, this is what the data looks like. Again, dark to begin with, we stimulate the tissue, we see release and reuptake of, in this case, dopamine. And again, all we're seeing here is the probe. All we're looking at is infrared wavelengths, and all we're seeing is the chemical and not the presence of the cells. But then we can average our signal over the field of view and just do some control experiments. So the stronger we stimulate, the more dopamine we get. We can add a drug called nomefensin, which is a dopamine reuptake blocker. It binds to these dopamine transporter proteins and it blocks dopamine reuptake. So at equal stimulation strength, in the presence of nomefensin, we can see that there is more dopamine that then resides in the extracellular space, and that it takes longer for that dopamine to clear, as we would expect from the mechanism of nomefensin blocking dopamine reuptake. So these are, again, just controls. One thing that was really important to especially our collaborators was that these probes be compatible with pharmacological agents. So when I say pharmacological agents, I mean drugs that are going to either be used as therapeutic agents, usually, again, targeting some of the ailments that I talked about at the beginning, depression, anxiety, et cetera, but also very broadly used for pharmacological-based interventions to study neurobiological mechanisms. So 2 examples of these pharmacological agents include sulfuride, which is a dopamine receptor antagonist, and quinprol, which is a dopamine receptor agonist. And again, dopamine receptors are kind of like the ears on postsynaptic neurons that are kind of tuning the sensitivity of that neuron to being modulated by that specific neuromodulator. One of the reasons this was not just a given is that a lot of times these molecules, sulfuride, quinprol, et cetera, will look very similar to dopamine, right? They're pharmacological agents that are meant to resemble the neuromodulator. So we were happy to see that in the presence of these pharmacological agents, we don't get a modulation in the fluorescence of our probe, and also that we can still detect dopamine in the presence, basically back application of these neuromodulators, all of this just in vitro. So this confirms compatibility of the probe with these pharmacological agents. We can, again, do just a control experiment showing that if we stimulate a brain slice that is just an artificial cerebral spinal fluid, we get the predicted release and decay kinetics. And if we add just BAF apply quinprol, we no longer get a signal. We can remove the drug and recover the original signal. Now, up until now, I've shown you data that is, again, an ensemble average of the field of view that we're able to capture with our imaging microscope. And of course, one of the advantages of working with a technique that gives you spatial resolution is that we're able to now kind of query individual kind of regions of interest or putative release sites within these tissues. And so just for this kind of pharmacological test, what Abraham then did was reanalyze the data, not by just looking at the broad scale modulation of how fluorescence or dopamine levels change in the addition of quinprol, which again globally suppresses dopamine release, but ask what does each release site do pre versus post addition of quinprol. And when we look at the data plotted in this way, what we see is something a little bit different, right? So here on the y-axis, we're just looking at the count, so these hotspots or release sites. And on the x-axis, we're looking at the fluorescence ratio. So before versus after adding quinprol, what does that one particular release site do? Unsurprisingly, most release sites are going to decrease, right, in their amount of dopamine release post-quinprol addition. But we see a good number of release sites that have the opposite effect. So they become almost hypersensitive to dopamine release or increase more dopamine after adding quinprol, which is an agonist. And so these data suggest one, endogenous variability, biologically driven variability, in how, in this particular case, dopamine release sites respond to pharmacological agents, and also suggests that there might be both pre and post-synaptic autoreceptor expression, right? Because we're seeing basically the opposite modulatory effect. And we see this consistently regardless of the drug. So there's always this spread that's concentration dependent in how these release sites are modulated by pharmacological agents. And so although this was, a project that now it's been published for a while, but that was meant to kind of validate the probe and its use in tissues, it taught us an important lesson that I'd learned here a long time ago, that biological systems can have many different underlying distributions that are important mechanistically to both understand and to inform therapeutic interventions, in the sense that similar populations can have the same mean or the same average, but have very different underlying behaviors. And so moving forward, we shifted to analyzing all of our data using kind of this component of the spatial information that we're getting, right? And when we no longer average our signals, but instead look at what each release site is doing, we can now count or quantify, for example, something like release site density, right? Within an imaging field of view where you have many neurons, how many of those are actually talking, right? Not on mute, but actually exocytosing neurochemicals. Of those that are talking, how loudly is each talking, right? And by using probes of different neuromodulators or 4 different neuromodulators, we can also understand, what language each of these spots is speaking with. And so moving forward, this is how we're doing our analysis. We're looking at the number of release sites in a tissue. Of those release sites, how much dopamine in this particular case is being released per release site. And when we start switching into different probes, what neurochemicals are being modulated differently as a function of some behavior or some disease. So one example for how we would use for example, our dopamine probe to understand how these features of modulation can change as a function of disease progression is by considering a neurodegenerative condition known as Huntington's disease. So this is a genetic disorder that's inherited and is categorized by these molecular events that occur as a result of this tag repeat that is overexpressed in human Huntington protein, and it causes expression of these Huntington aggregates then cause demise of medium spiny neurons, which modulate motor centers in the striatum of the brain. And because these motor centers are also very heavily innervated by dopaminergic neurons, we hypothesize that the dopamine pathway might somehow be compromised in these animals. So this is basically a transgenic line of animal known as an R62 model of Huntington's disease. It basically has this human Huntington gene cloned into it, so it degenerates quite quickly within a 12 to 13 week time frame. And again, what we're looking at here is the central question being, how does dopaminergic release change as a function of disease progression, both in terms of the number of unique dopamine release sites, but also how each of those release sites might be changing its ability to release dopamine as a function of disease progression. And what we do for this and other studies is to look at behavior or some sort of symptom separate from the kind of chemical activity, right? So in the case of Huntington's disease, one assay that we do to measure quantitatively disease progression as it pertains to motor symptoms is with what's called a rotarod assay, where age-matched littermates are genotyped shortly after birth and then split into cohorts for Huntington's disease positive versus wild-type animals. And then they are assessed for rotarod performance. They're basically on this rotating rod, and they can stay on fairly well if they have the musculature to do so. But then as disease progression occurs, they start falling off the rod more quickly, and we can quantify how well, basically, the time they spend on the rod. We can then sacrifice those animals after assessing the rotarod performance at 3 different time points: pre-symptomatic, where motor symptoms start to occur, and then when cell death is evident. And then take those tissues, which again preserve the intactness of the cellular architecture, label those tissues with our probe, and then look at how dopaminergic signaling varies as a function of disease progression. All right, so again, looking at this example video, what we're looking at here is no longer this ensemble average video of how much dopamine release might change, but rather looking at those metrics of the number of individual release sites and how strong each of those release sites is turning on, which is a proxy for the amount of dopamine at each release site. So the first data set I want to show you was kind of this comparison between the ensemble average data set and the individual release site data set. Right, so both of these are coming from the same cohort of animals, where blue is wild type and orange is Huntington's disease, as a function, again, of disease progression on the x-axis. So unsurprisingly, as animals age or get more sick, depending on which cohort they're in, we see a gradual but slight decrease in the amount overall of dopamine that we can measure per field of view, right? So this is an average measurement that's a proxy for the dopamine concentration. So we're seeing a depletion of dopamine in Huntington's disease as a function of disease progression. But then if we look at the number of release sites, here we can much more clearly see, especially at the 12-week time point, a much more strongly compromised metric, which is that we're seeing many fewer release sites in the advanced disease stage than in the wild type. So motivated by this kind of very initial pilot data set, we then wanted to start looking more in depth at the mechanisms that underlie this global, again, suppression of dopamine as a function of disease progression, because you can imagine that there would be many different ways that dopamine could be depleted in a disease model like Huntington's disease. It could be that the synthesis of dopamine is compromised and therefore there is exocytosis, but those vesicles are empty. It could be that the exocytosis process itself is compromised, or it could be that dopamine is being decreased, again, globally, but is being compensated for by an increase in dopamine release for fewer release sites, right? Many different mechanisms. So the one we're testing here is with regards to the ability of cells to exocytose dopamine in the first place. And the way we do that is actually quite simple. We just change the extracellular calcium concentration to make cells more or less sensitive to dopamine release. So just looking at the 4-week data here, the color scheme is the same. So blue being wild type, orange being Huntington's disease. As a function of calcium concentration, as expected, both of these trends are roughly linear. So the more calcium, extracellular calcium you have, the higher the chance that a cell will exocytose dopamine. And that's basically no surprises here. At the 12-week time point is where we start seeing deviations from linearity, right? So the wild type animals still follow this predicted kind of linear trend in terms of sensitivity to dopamine release. But in the Huntington's disease case, we now see this suppressed ability to release dopamine as a function of release site or hotspot number. And this suggests that in dopamine or in Huntington's disease, we start seeing a compromise in the ability of voltage-gated calcium channels to be sensitized essentially to exocytosis of dopamine. The second bit of data that was interesting to us was that although it did not reach statistical significance, for physiological and high calcium concentrations, we almost see an inverse relationship where it's actually the Huntington's disease animals that are more likely to release dopamine, suggesting a compensatory effect early in the pre-symptomatic time points that then are compromised much more in late disease stages. Yeah.
How do you know that you're looking at a synapse?
Yeah, so that release site and synapse are, in my mind, two different things, right? So if we were to want to claim synaptic release, we would need to measure the release size and ensure that that's the case for a single neuron. So what we have are data from the BNA lab now on these two-dimensional cultures showing that the release site and the volume transmission is on the order of 3 microns. And so because we're able to also measure 3 micron spots in our images, then we can at least associatively say that those are, putative release sites. Are they individual release sites? That we cannot say without doing the corresponding microscopy where we're looking at the cell bodies and the cell projections as well. But because they're on the same size, on a physiological level, it does seem like what we're picking up are at least individual release sites, if not synaptic. Yeah. So one additional kind of metric that we've been using recently to start looking into these mechanisms is this concept of release site fidelity, right? So if you monitor the same tissue over multiple attempts at signaling, which in our case is multiple stimulation events, it's, again, feasible that there would be similar release probability for that very first exocytosis event, but that the more you stimulate the tissue, you might have a decreased fidelity in release. And so what we then do is we look at entire field of view and we track the same release site over multiple simulations. If it's a high fidelity release site, we consider that a release site that releases 3 times in a row. If we stimulate 3 times but only get released twice, that's a medium fidelity release site. And if we stimulate 3 times but only see release once, that's a low fidelity release site. And so we can do this in the presence of pharmacological agents that are supposed to, again, increase the sensitivity of our neurons to dopamine release. And what we see, again, is in this comparative manner between wild type and Huntington's disease, is that there's less of a difference in that very kind of low fidelity release site regime, right? Where cells, both in Huntington's disease and wild type, are likely to release dopamine at least sometime. But it's in the blunting in dopamine release happens with these subsequent release events, right? So again, with the analogy of the person trying to speak on mute, in Huntington's disease, it's not so much that there's a compromisation of of dopamine synthesis per se, but it's in the release machinery that we're seeing blunting of dopamine release. Yes.
I had a question on the previous slide. Do you have any sense for if there's a refractory period, I guess, for cells in the fidelity 3
Yeah,
group?
absolutely. Yeah, absolutely. So there are a lot of factors that drive this refractory period. What we do with these repeat stimulation experiments is to let the tissue rest for a good 15-20 minutes in between stimulation events. So it through control experiments shows that more than allows cells to recover and even reuptake any extracellular dopamine that they would want to recycle. So we're quite confident in being able to measure these release fidelities as a compromised exocytosis machinery. Good question. All right, so a couple years into the project, we ran into somewhat of an issue in how we are generating our probes, and that is the way that we generated our dopamine probe and the way that we were trying in that time to generate probes for other neuromodulators was through this serendipity or luck-based process of screening polymers for which we didn't know if there would be any affinity for the analyte against an analyte library. And that's, of course, very difficult to do if you want to generate a new probe. With a purely luck-based approach, it could take forever, essentially, to generate a new probe if we were unlucky in that initial screen. So a former postdoc in the group, now a faculty member at Pusan National University, Sanghua Zheng, took on this project of questioning the concept of being able to evolve molecular recognition. Now, because in the case of dopamine, and we hypothesize other molecules, there might be molecular recognition that's achievable with nucleic acid polymers, the concept here was whether we could develop molecular recognition on the surface of these semiconductors for a target that had eluded us through screening, like serotonin, based on a large random library of nucleic acid polymers that we would competitively complex to the surface of these semiconducting carbon nanotubes in the presence of an analyte that we want to develop molecular recognition for. So it's not unlike aptamer generation, except here we have this third entity, the carbon nanotube, that is serving as a template for the structure of the nucleic acid that would go on to perform the molecular recognition. We unfortunately cannot take aptamers from the literature, for which there are plenty, for neuromolecules and just put them on carbon nanotubes because there's a very strong affinity through sp2 bonding for any nucleic acid base and the carbon lattice of the carbon nanotube. So we have to develop this molecular recognition post-template to the carbon nanotube. So the concept, though, is very similar, right? So we adsorb these polymers onto the surface of nanotubes in the presence of serotonin. Presumably, there's a subset of the population in terms of sequence that will bind both the nanotube and the analyte well. We can remove unbound sequences and then collect the bound sequences, amplify those sequences with PCR, and then sequence them. So we know with each round of evolution what the population or distribution of sequences is. And we can do this over multiple cycles. And because, again, we have this kind of trimolecular interaction, we also run a control in which we competitively complex the same library with just carbon nanotubes alone, right? So here we're looking at sequences that might be good binders to the carbon nanotube surface, but not necessarily for serotonin. So we can remove those sequences from consideration when looking at the sequences that we want to build our probes from. And then we can do this over any number of rounds. And because we have the sequencing data from every single round, we can look in principal component, like a non-dimensionalized space. So do basically PCA analysis on the composition of the control library with the experimental library. When we start seeing divergence in PC space, we hypothesize that there's something about the presence of serotonin that's driving that selectivity. And then we can actually look at what those top sequences are for that round of evolution, synthesize those probes, and test for response to serotonin. The data from the consolidated experiment I just described are here. So as a function of experimental rounds around 3, 4, 5, and 6, and this is the control where we don't have any serotonin, we see a gradual increase in the fluorescence response pre versus post addition of serotonin, where the best performing sequence here is this E6 number 9. So this is a basically the ninth most common sequence in the sixth evolutionary round. Compared to the control library, where we do see a small kind of gradual increase in the fluorescence response of these nanotube nucleic acid complexes. That is unsurprising because we are still evolving for enhanced molecular absorption of these sequences to the nanotube surface. But we've all but switched to this basically process of generating new probes because of how efficiently we can do so, and that we can then use these data sets. I'm not going to show any of this work, but in collaboration with Leila Vukovic's group, we've shown that we can use now machine learning to kind of predict sequences that might not have actually come up in our experimental data sets to build even more responsive sensors. Yeah.
This approach, so you are selecting for, I guess, selectivity, I guess.
That's right.
But not
Sensitivity.
specificity.
That's right.
Specificity, is that right?
That's right. That's right. So that's one drawback, is that when we used the old screening approach, we had the selectivity screen built in, right? Because we were always looking at what the spectral response was for those polymers. The rate limiting step in the serendipity-based approach is getting any hit at all. So this gives us that any hit. Rarely do we use the sequence directly because of the selectivity issue, but then we can iterate on that sequence to then get to the both sensitivity and selectivity needed to be used in a brain region of interest. We can also do a counter screen, so we know which sequences are responsive for all of these competing neuromolecules. We only need to do that once, because once we have that information, we can just exclude those sequences from consideration when we're basically doing the selectivity component of probe development. Yeah, and so we've used this approach in addition to a few other approaches to generate a probe for oxytocin. This is something I was really excited about because oxytocin it's a neuropeptide, so it's a larger than a classical neuromodulator, peptide-based signaling molecule that's implicated in a lot of really interesting but subtle behaviors. And so through a collaborative effort within the lab, using the selects approach and also a few other, again, modifications to enhance selectivity and sensitivity, we've been able to develop an oxytocin sensor that not only is nicely responsive to oxytocin, but does not respond to vasopressin, which is really important because in many oxytocinergic regions of the brain, there's a competing neuropeptide pathway that effluxes vasopressin. These are molecules that are very similar in structure, differing by really only 2 amino acids. And so the ability to distinguish between the two was important to us to claim oxytocin specificity. And so with this probe, we've started a new kind of area of research in the lab that I'll close the talk with, and none of this data is published, so this is, I'd say, the area where we'd most love to get feedback on. But oxytocin is, you might have heard of it as kind of like the love hormone or the love peptide. It's implicated in pure bond formation, broadly speaking. And it's been studied quite heavily in the context of what are called mate or reproductive relationships. So this is really kind of simple but beautiful experiment showing that for voles, which are quite social animals, in a mate relationship, right? So this is a reproductive relationship. The contact time, the amount of time these two animals choose to spend with each other, increases after infusion of oxytocin, but not after infusion of artificial cerebral spinal fluid, right? So just exogenous administration of oxytocin causes an increased preference for a known romantic partner. Some newer data out of the Rob Frumke's group at NYU did this really nice experiment looking at the effects of oxytocin in maternal offspring relationships. And so mice will have pups, and these pups might start crying if they're left alone, and then a mother will usually retrieve a crying pup, both her own and other crying pups, if she hears them. But virgin mice won't have this retrieving behavior. So virgin mice will basically leave a crying pup by itself. And so the experiment that the Frumke lab did, this is by Bianca Jones-Marlin, was to either infuse oxytocin or optogenetically stimulate oxytocinergic neurons, and compared to a saline control, what they saw was an increase in the retrieval percentage by virgin mice, right? So essentially what this is saying is that just activation of oxytocinergic neurons, which presumably causes exocytosis of oxytocin, or intravenous administration of oxytocin causes virgin mice to learn this kind of pop crying retrieving behavior, right? And this is something I think I experienced myself in a very kind of different setting where I travel quite a bit. And so before having children, I would hear, a crying infant on a plane, and my instinct was just to put on headphones, ignore it, and work on a new grant or something. But since having kids, I hear a crying infant on a plane, and it's just, it's kind of heartbreaking. And I have a lot of empathy for both the child and the parents for that matter. But there's, certainly something going on there in kind of mammalian reproductive relationships as it pertains to oxytocin. Now, one area where oxytocin is far less studied is in selective peer relationships. So everything I've talked about now are relationships that are very strong in the sense that they are central, right? So reproductive relationships can be thought of as a partnership or a marriage. Similarly, maternal offspring relationships. But arguably peer relationships, which are closer to friendships, are much more numerous and very important for supporting human health. Peer relationships are also thought to be important to study as it pertains to autism spectrum disorders, because oftentimes it's these peer relationships that tend to be compromised for individuals who are on the spectrum. There are also have been a few small scale trials suggesting that intranasal administration of oxytocin can alleviate autism symptoms in children. But then a very much more recent large randomized clinical trial showing basically a null effect of oxytocin administration and alleviating autism spectrum disorder symptoms. So there's just a lot of controversy in the field, specifically as it pertains to the role of oxytocin in the formation of friendships and aberrations thereof. So we started this study on a new organism called basically these prairie voles. And the reason we choose prairie voles is because they are capable of forming selective affiliations. And we can measure this doing something called a partner preference test. So what we do is we have a vole, or rather two voles, and we allow them to interact for a certain predetermined amount of time, right? So it could be a lifetime, it could be a short amount of time. And then we place one of the partners and we tether them to one side of a box. We tether a stranger vole to the other side of the box. And then we have basically the friend of this animal in the middle. And we measure the amount of time that vole, the focal animal, chooses to spend huddling with its known partner or versus a stranger. And again, these are all non-reproductive assays done with the same sex partner. And just to give you a reason for why it is that we work with voles specifically, these selective affiliations and non-reproductive partnerships are only exhibited in about 9% of species. So friendships that you and I form are actually quite rare. So there are very few animals that we can use to study this. So here we're just looking at partner preference tests. So running this assay with just mice versus voles. So you can see a prairie vole will have a very strong preference to huddle with a known partner versus a mouse that doesn't really seem to care how much time, if at all, it spends huddling with either. So again, intuitively, I personally would, if I had to huddle with somebody, I'd much rather huddle with a known person than a stranger. And that intuition is, again, something that surprisingly very few animals have. All right, so this is a project that we started with Annalise Beery in integrative biology at Berkeley. And what we're using as a model organism, in addition to these voles, is a transgenic vole that is basically a receptor null mutant. So it does not have the protein receptors that can basically hear oxytocin. And so essentially what we're doing are comparing wild-type voles to voles that do not have the capacity to signal through oxytocin. And at the start of this project, we had a lot of questions, including, how is it that oxytocin receptor null mutants are going to change in their behaviors? Are there any changes to oxytocin either synthesis? So do these voles have any oxytocin at all? If so, are there any signaling aberrations, and where in the brain are these signaling aberrations found? So the first thing we did was this partner preference test. So again, running that test that assays whether a vole prefers to spend time with a stranger versus a partner. And the first test we ran was a lifetime partner. So this is your best friend since childhood. Do you prefer to huddle with your best friend or huddle with a stranger? And what the results are that both the wild type voles and the ones without a functional oxytocin receptor both prefer to huddle with their lifetime friend, which is in a sense good news because it suggests that there's still the capacity to form that partner preference, even without a functioning oxytocinergic circuit. However, if we redo the test, instead of using a lifetime partner, so this isn't your best friend since childhood, this is somebody you met at the coffee shop yesterday, now we see a difference between the wild type voles that prefer to spend time with their coffee shop friend, despite being new, versus the receptor null mutants, or the knockouts, that don't seem to have any statistical preference between huddling with a stranger versus a partner. The good news is that if we now take kind of an intermediate approach to the test and rerun the test with a 7-day partner, so this is somebody that you met at the library and have been keeping in touch with since last week, now we can see a recovery of preferences for that known library friend over a complete stranger, despite, again, this completely blunted oxytocinergic pathway. So these data, again, are promising in the sense that there is still the capacity to form selective non-reproductive relationships without a functional oxytocin receptor. But we were interested, again, in looking at the underlying oxytocin dynamics in the same kind of metrics that I already motivated before by looking at release sites and how much oxytocin is released per release site. And again, what we're quantifying here are metrics including number of release sites and oxytocin release per release site. But now we're using our oxytocin probe, and so we're just measuring a different neurochemical. And so these data are all extremely preliminary, very small sample sizes so far, data acquired by a new postdoc in the lab, Natsumi Komatsu, who is a Schmidt Science Fellow. Yeah, again, the questions here being, are there differences in oxytocin signaling? Is there oxytocin at all? And where regionally does this, or do these differences start to arise? So we looked at 2 different brain regions, the nucleus accumbens core and the central amygdala, both of which have a very high, in at least wild type, expression density of oxytocin receptors and are centrally implicated in peer affiliations or the formation of peer bonding interactions. So question 1, do oxytocin null mutant voles have any oxytocin at all? The answer is yes, they do. So here I'm just showing the analyzed data from these videos, comparing wild type animals to the receptor null mutants. So the receptor null mutants do have oxytocin, both in terms of the integrated amount of oxytocin per field of view, the number of individual oxytocinergic release sites that we're able to measure, and how much oxytocin is released on average per release site. But in all 3 metrics, we're seeing, again, a decrease, a statistically significant decrease in the amount of oxytocin, release sites for oxytocin, and the amount of oxytocin released per release site. So this clearly suggests a blunted ability to signal through oxytocin in the receptor knockout voles.
Do you know where it's made? In the...
Yes, in the VTA. So that's basically below the hypothalamus is where the soma are that would synthesize.
So they know the pathway.
Yes, they know the pathway. And oxytocin is not reuptaken. It is very quickly degraded. So it has a slightly different mechanism also post-signaling of requiring synthesis for its presence to be exocytosed. Yeah. So it was actually surprising to us that, one, there was oxytocin because it's expensive to make and it's something that can be very easily regulated at the point of synthesis. And 2, in the community, it was thought that there would actually be an increase in oxytocin generation that could then signal through vasopressin pathways because the receptors for oxytocin and vasopressin often confuse the 2 neuromolecules endogenously and are found in the same brain regions. So the suppression of oxytocin as opposed to an increase in oxytocin suggests that there are other neurochemical pathways that are compensating in these receptor null mutant animals. Yeah, good question though. Yeah, so regionally specific, question mark. So now we're looking at data in the central amygdala, and we see no difference by any metric. So overall global amount of oxytocin, oxytocinergic release sites, or how much oxytocin is being released per release site, don't differ at all, despite also being a brain region that's centrally implicated in peer relationship formation. So these, again, strongly suggest regional differences in how oxytocin signaling deficits are emerging in the brain, and we have yet to to scratch the surface of why this regional difference might be. Any sex differences in oxytocin signaling? We've only had the chance to analyze wild type data so far, but we don't seem to observe any sex differences in wild type bulls in terms of, again, these oxytocinergic bluntings. As you can tell, we have a very small sample set so far. So we're going to reanalyze this data with both wild type bulls and also the receptor null mutants once we have the statistical power to do so. And lastly, kind of piggybacking off of Zan's question is that it's very likely that, and it's been hypothesized in the field that whether it's a neurotypical behavior, whether it's a disorder, that it's multiple neurochemicals simultaneously at play, right, that are driving these behaviors or these disorders. And so it's still impossible to this day to measure multiple neurochemicals simultaneously, either from an imaging perspective. You can always take a sample of CSF and then measure it offline, but in terms of real-time kind of data, to be able to measure multiple neurochemicals at once. And so I foresee the next 5+ years of the lab's efforts more heavily focused on enabling this technology for simultaneous neurochemical imaging, so we can start mapping neurochemical release across not just brain regions, but with multiple neurochemicals at once. And I don't have any data to show you, but we do have a study looking at basically repeating this whole oxytocin study, but with our dopamine probe. And we basically see a mirror image in terms of signaling deficits when it comes to dopamine. So dopamine signaling compromised quite heavily in the central amygdala and not so much in the nucleus accumbens. Again, strongly suggesting an interplay that has as of yet to be determined between multiple neurochemical signaling pathways and the importance of being able to measure all of them at once. So I'll close the talk just by, as promised, highlighting some of the work that's being done in a few different alumni groups. So the Bayene lab at the Janelia has built these absolutely fantastic microscopes that are able to do the dream experiment of looking both at the morphology of cells in tandem with neurochemical release. So what Abraham's data is showing here are basically primary neurons that are labeled with GFP and have been grown on these dopamine sensitive films. So these 2D arrays that are sensitive to dopamine. And what Abraham is able to measure is both with stimulation, but also with just spontaneous or endogenous release events from neuronal projections or dendrites. What's really beautiful about Abraham's data, especially the one that looks at multiple neurons together, is that he's seeing that release events don't necessarily come from dendritic projections alone. So he's seeing somatodendritic release and even the capacity to release from the soma, which previously was not really thought to be canonical part of of neurochemical signaling pathways. So a lot of Abraham's future work is kind of looking essentially at where neurochemical release happens as a function of all of these different neuronal architectures, and in my opinion, kind of redefining how it is that we think of neurochemical communication, not happening just at these classical dendritic synapses. Another alumnus, Rebecca Pinels, who's starting her lab at Stanford and recruiting, so if anyone's interested, please reach out to Rebecca. Looking at the use of these materials or these probes in human-derived brain organoids. So one of the advantages of working with a synthetic probe is that it doesn't need to be genetically engineered to be expressed in all sorts of different species. Of course, human tissues being a bit of a holy grail of where we want to study human diseases as opposed to engineering them into model systems. And so Rebecca's lab is looking at neurochemical signaling and aberrations thereof in three-dimensional brain organoids as it pertains to Alzheimer's disease. And with that, again, thank you so much for the invitation. Happy to share some of our lab's research with you today. Big shout out to my group, those that supported our work, our collaborators that I've highlighted along the way, our funding sources, and yeah, happy to take any more questions.
And then how much time do we have for questions? It's a little past.
Oh, until somebody comes in.
Okay. All right.
So I have a quick question. So do they know much about, or the structures of some of those receptors that they've been determined and how they differ between the different
That's a good question. Yeah, I think Paul's maybe a good person to look into this. very little is known about the positioning, the expression, the location of these receptors, and they're recycled on a relatively short time scale, right? So it can, even that is, I think, somewhat unknown, but it's not like it takes lifetimes for these receptors to express and localize into the cell. They're expressed and then they're kind of ready to be inserted into the cell when the cell needs to be more excitable or less excitable, more or less
When
sensitive.
they're diseased, is there a mutation perhaps in the transporters? Because you mentioned, looks like it's the release.
That's right.
And so that would be some indication. If you could
That's right.
see, they could isolate the receptors for the diseased animal from a healthy animal.
That's right. I don't know how much of that is known either. And it depends on each neurochemical. So in terms
Sure.
of, dopamine, there are, dopamine D1, D2, D3, D4, and all of these isoforms are so structurally similar that it's difficult. You can't use them in histochemistry. The antibodies for them are horrible. So you can't really tell them apart. For oxytocin, it's much more straightforward. I think the challenge there is that we haven't yet, at least in our hands, we haven't yet conclusively determined that what's causing the blunted release is a receptor sensitivity issue. So it could be on the synthesis side. It could still be one of many things that we're going to do the quick assays for before diving into or committing to looking at the receptors in more detail.
Very nice.
Questions? Maybe some students. Right, you want to go first, Zipper?
Yeah, I have two questions, so I'll start with the first one. So I'm curious, the Huntington disease study, is it a way to maybe have two different or three different maybe sensors at once, and kind of like how distinguishable those sensors are? Basically, like, have you looked at the ratios of the different modulating neurotransmitters?
Yeah, that's again, that's kind of like the longer term plan. One of the challenges for using our probes as is now is that they're a multi-chirality probe, right? So we can do experiments like the oxytocin work with an oxytocin probe and then redo the whole experiment with wolves with a dopamine probe and then look at what happens with oxytocin quantitatively and compare that with dopamine quantitatively. And because we're looking at metrics like release site density, those are actually nicely comparable. But the issue is that we can't take the data simultaneously, right? So I would love to see, okay, dopamine release site here and oxytocin always co-releases with it or never co-releases with it. That would be beautiful. So what we're hoping to do is to separate. So these are how the semiconductors the nanotubes are synthesized. They can laboriously be separated out into individual chiralities, chiral vectors or colors, essentially. So they have unique excitation emission spectra, and then functionalize each with the polymer that corresponds to dopamine for yellow and then to oxytocin for blue, and then use them in a multicolor still infrared microscope. We're still quite a ways away from being able to do that both on the chemistry front and on the microscopy front.
Yeah, and I have a follow-up question. I mean, it's about the second project.
Yeah.
I'm just curious. I'm not sure. I don't do animal studies, so like I'm not really sure about this. But are there any measures in place to control for mate gender preferences of the animal? Like, is there an underlying assumption that all the prairie wolves are heterosexual, or like how can you control for that?
Yeah, that's a good question as well. So right now, the way that we take data is so these partner preference tests are always done with the same sex, but we assay both sexes. So we separately look at females and separately look at males, and then we analyze the data separately. And so far, we haven't seen any differences between males and females in terms of all of the ROI analysis, et cetera. So then we can pool the data once we confirm that there's no differences. And if there are differences, we just double our sample size to include both sexes separately. So that's the way we approach it, practically speaking. I don't know enough about the field to know about whether there could be a romantic analogous kind of element to same-sex partners, the way that we're assaying like friendships now. So yeah, I'd have to learn more about that before confidently answering, but it's a test that's been used for about 2 decades to measure these very specific types of relationships.
I think you touched on this a little bit, but do you know what is there a way for you to track the fate of the chemical messengers? Because you see they go away, some of them are reuptaked, and you're saying oxytocin is degraded, but do we know all of these, like what percentage is being taken up by the postsynaptic neuron?
Yeah, we don't know that. We do have a separate probe that's based on Raman imaging that looks at intracellular imaging of these neurotransmitters and neuromodulators. But we cannot yet, that's all visible spectrum, so we cannot yet use it in conjunction with this specific assay. So no, we don't know. The decay constants, right, the reuptake component of the curve is a combination of diffusion and reuptake. And indeed with dopamine, where we know that transporters are involved, we see a much sharper reuptake curve than with oxytocin, where it's 100% diffusion because there is no reuptake. So we can look at comparative measures of that decay constant and make inferences based on that, but we cannot say, yeah, this particular molecule was
That had X amount.
Exactly.
Okay.
Yeah.
Thank you.
More questions in the back?
Thanks for your talk. I thought your studies on with the bulls was really interesting. Have you looked at like whether the friendship affiliation behavior is like dependent on like the age of the bull, when they become friends, or also like what's their environment as they're growing up, like if they have age mates for a long time, or if they're like moving around or by themselves.
Yeah, we would love to do all those. We have not looked at that. All of these are with adult mice, but we have started putting in new elements. So we have an addiction study that I didn't talk about, but we're then incorporating food insecurity into our addiction study and seeing that if you grow up with a food insecure upbringing, then you're likelier to become addicted to cocaine. And we show that autoreceptor expression as measured through the lens of neurochemical imaging is different. So mechanisms of addiction being different. We haven't looked at all of that in terms of social relationships or friendship formation, but I have a hunch that'd be very different. And what's interesting is that for voles, where oxytocin receptors are expressed is extremely dynamic. So it's unlike dopamine, where you kind of know what parts of the brain throughout life you're going to find high dopamine receptor expression densities. For oxytocin, that changes drastically during development. So the brain regions implied in forming friendships are probably much more dynamic than those that are involved in like learning or memory.
I have a quick question.
Okay, go ahead.
Can you apply this in vivo in live animal?
I was gonna ask.
Yeah. Yes, so Abraham can do that. We can either do one of two things: inject the probe the way you would inject a any sort of other neurochemical reporter or neuro probe, and then use fiber photometry. So drop a fiber and then get the signal out. The reason I'm not as enthusiastic about going that way is one, in the past few years, there have been protein-based probes for neurochemicals that already really nicely give you fluctuations in neurochemical signals as a function of real-time behavior in vivo. But it compromises the spatial information. So I've yet to see any other kind of, use of those probes to measure more nuanced differences in neurochemical signaling. So if we use that oxytocin probe with a fiber photometry tip to measure partner preference formation, we actually don't see any differences in oxytocin. It's only once we start looking at release sites that we see differences between the two.
Do you face particle aggregation issue with those particles?
Yeah, thankfully we don't. So when we synthesize the probes, one of the first filters that we use is colloidal stability, and that depends on the binding strength between the polymer and the nanotube. Because what causes aggregation is usually kind of pi annihilation of nanotubes that are not well suspended with polymers. So we only work with polymers that essentially have like a zero off rate, even in cerebrospinal fluid. One of the reasons I think these probes work well is because we're working in the extracellular space of the brain, which is devoid of clotting factors. Any type of nanoparticle, I think, is susceptible to colloidal instabilities when we start working in like blood or fibrinogen clotting factor rich media. So I think we're just fortunate that we are working in probably one of the cleanest parts of the body.
I can ask a two-part question related to that point. So just thinking for this audience in terms of how broadly applicable this technique is. So the part one is like, what are the everything you've shown so far is imaging outside the cell, right?
That's right.
So what are the part one is what are the prospects of actually imaging inside the cell? I guess part two, which is related, is how broad is the palette of analytes that you can basically, or is there something specific about neurotransmitters that make them particularly amenable to this technique, or can you really kind of design it for any kind of analyte that you think might be inside a cell?
Yeah, intracellular measurements, I am also hesitant about for a similar reason as the last answer, that the intracellular environment is complex, it's dense, it's dynamic. And so my selectivity screen, I have less confidence in whenever I'm working in an environment that I don't have good control over what's there, right? So for all of these selectivity screens that we do, we have microdialysis data showing that those are the molecules that are present in that brain region, and we can be confident that the response is therefore attributable to just that one neuromodulator. Once you go inside the cell, that becomes almost infinity. So the secondary metabolites or small molecule analytes that can perturb the optical response of the probe. So we do have some Raman-based probes that work well for intracellular imaging of neuromodulators. But I don't think that these probes would be the best suited for that. I do think that they're especially well suited to study neuromodulators because of this diffusive activity. So we haven't yet, but could develop probes for something like GABA or acetylcholine that are ligand-gated ion channel-based neurotransmitters and supposedly only kind of shuttle through the synapse, but not really outside of the synaptic cleft. That is still hypothetical, so it would be really nice to see if that's actually the case. I would anticipate that if it was really just synaptic transmission, that we would see very, very small, very fast signals, as opposed to these diffuse, longer-lived signals that we're picking up with oxytocin and dopamine.
You don't think there's anything in terms of the chemical structures of the neurotransmitters that you're looking at that make it particularly
I see.
difficult the binding to these sensors?
I don't think so. There's not, I mean, dopamine is a redox active molecule. So when we had trouble screening for serotonin, we initially thought maybe it's because of it's not a redox active molecule or oxytocin, but there doesn't seem to be no physiochemical property of the molecules we've successfully measured so far have had any obvious correlation. So I think the process is generic enough. The biggest thing being the selectivity. So I want to be confident that what I'm measuring is the thing that I'm measuring.
Let's thank Markita again for the